Assalamu alaikum. Good morning. I am Dr. Mdassar Hassan. I welcome all of you in 8th International Conference on Applied Zoology 2025, organized by the Applied Zoological Society of Pakistan in collaboration with University of the Punjab Lahore and Government College University Faisalabad. Here we are going to start our first session, which include the talks from the field of biodiversity, entomology, ecology, and fisheries. So let's start our talk with the first speaker, Dr. Omarhan Durrani from Karadeniz Technical University, Trabzon, Turkey. He will talk on the spatial growth modeling of Atlantic bonito in the southern Black Sea. Please, Dr. Omarhan Durrani.
Hello, good morning. My name is Umarhan Durani, working at Karadeniz Technical University, which is located at Eastern Black Sea coast near to the Georgia. In this presentation, I will present a part of work on Atlantic bonito, which was done under a project funded by our national funding agency. Atlantic bonito is a commercially valuable pelagic predatory with a broader distribution across the Atlantic Ocean, Mediterranean Sea, and the Black Sea. It supports economically significant fisheries throughout its regions, particularly in our regions, the Black Sea, where it undertakes seasonal feeding and spawning migrations. What is a fish stock? Before explaining the findings of my study, I will try to briefly explain what does a fish stock mean. In fisheries management, a fish stock is a crucial concept. It refers to a subpopulations of a particular fish species that is treated as a single management unit. This means that the scientists and the fisheries managers, assume the population's dynamics like its birth, death, growth of that stocks are largely independent of other stocks of the same species. There are various methods that are used to identify fish stocks of a fish. There are morphological markers, molecular genetics, and otolith chemistry. These three are very famous in fisheries management. My work was on morphological markers, so I will try to explain the morphological markers part. For stock identification of Atlantic bonito to investigate their stocks in our sea, in the Black Sea, we sampled this fish from three different regions, which covered the eastern Black Sea, like Trabzon, where our university is located, the middle Black Sea, Sinop, and the one was from western Black Sea, which is the sampling station was located in the Istanbul. We took fish images for geometric morphometric analysis, and we extract the otolith to use them for shape analysis, like in elliptical Fourier analysis of the otolith, to determine different stocks of our Atlantic bonito. As you can see that the morphological markers have successfully identified two different stocks of this fish. We sample the fish from three stations, Trabzon, Sinop, and Istanbul. The fish sampled from eastern, middle, like Trabzon and Sinop regions, were found to be significantly similar, like they could be considered as a single stock, while the fish sampled from western Black Sea, from Istanbul, they were found, or they showed considerable significant differences in both otolith shape and body shape. So there are two different stocks of this fish in our sea. Since we have found that there are two different stocks of this fish based on body shape analysis and otolith shape analysis, for the first time we tried to find out whether the growth rate could be used as a stock identification markers or not. So for this we identify the fish age from their otoliths and then use the XY plot and apply the four different growth models. Van Bertalanffy and exponential 3p were found to perform very well based on R2 value, R square value. So, for further analysis to compare the growth rates of our fishes, we use the Van Bertalanffy growth function since it has been widely used in fisheries. In the second table, you can see that the growth rate of Western Black Sea, the Atlantic bonito from Western Black Sea, has considerably different growth rates than both Middle Black Sea and Eastern Black Sea. Again, we use the ANCOVA regression equation to find out to compare the slopes, the growth slopes of our Atlantic bonito. And again, we have found similar results like the results which was provided by otolith shape analysis using the elliptical Fourier analysis and that provided by body shape analysis using the geometric morphometric. So we can say that growth rate can also be used as a stock identification markers, which is a good finding. Thank you so much for your time. Have a nice day.
Thank you very much, Dr. Umhan Durrani, for your valuable talk. Now we will move to our next speaker, Dr. Patricio R. from Chile. He will talk on null models for understanding intertidal decapod communities in North Patagonian Beach, Chile. Please, Dr. Patricio.
Hello, it is the presentation entitled New Model for Understand Intertidal Decapod Communities in North Patagonian Beach, Peyuin, 41 South Latitude, Chile. The speaker is Patricio de los Ríos Escalante from Catholic University of Temuco, in Chile. In this slide are mentioned the co-author of this study, Patricio de los Ríos Escalante, Eliana Ibáñez Arancibia, Farhan Albori, and Carlos S. As introduction to Chile North Patagonia, 38 and 41 south latitude, is characterized by the presence of coasts with marked wide inflow exposure and low productivity conditions that does not allow high species richness. Finally, in Patagonia south of 41 south latitude, the presence of inner seas and relative low human intervention allow the presence of high crustacean diversity. In order to study communities, one of the focus areas is the use of neural models, which are based on the previous condition that community is either random or there is not a structure. These models are more robust than another kind of statistical analysis. A neural model is applied in this study in order to better understand the ecology of intertidal decapod community on northern Patagonian beach. In material and method sections, the study site is as on the beach with small rounded stones called Peyuin Beach. This is the geographical coordinated and the map and a photograph of landscapes at 4 kilometers at south of Puerto Montt in a semi-rural zone that was visited in 24 February 2020 during low tide. As data collection, first species identification was done in situ, based on literature descriptions, and taxonomic identification was confirmed by worms. Random quadrants, 10 by 10 centimeters, were released in study sites during low tide. It was considering this zone of quadrant on the basis of fast movement of littoral dipoles, and in this size quadrant will be more adequate for fast movement species and the irregular conformation of the site around the rocks. Manually entered data were recorded in a field copybook. As data analysis a community structured by competition when C-score is significantly larger than expected by chance. Consequently, we compare concurrence patterns with null expectation by simulations using statistical neural models, fixed. In this model, the row and column sums on the matrix are preserved. Thus, each random community contains the same number of species as the original community, fixed column, and each species occurs in the same frequency as the original community, fixed row. Whereas in fixed equilibrium algorithm, only the row sums are fixed and the columns are treated as equiprobable. These neural models treat all samples columns as equally suitable for all species. In fixed proportional algorithm, the species occurrence totals are maintained in the total community, and the probability of that species occurs in a sample is proportional to the column total of that sample. The neural models analysis were performed using software R and package EcoSim-R. For second-year models, is a niche overlap analysis, and individual matrix was built with rows and columns represented the species and site respectively. This matrix was used to test the niche overlap significantly differed from the corresponding value under the new model's hypothesis, random assemblage. These analyses were applied to data from the second field period and were based on Bianca index. This model showed the probability of night sharing compared to the niche overlap of theoretically simulated community. The niche amplitude can be retained or reshuffled. When it is retained, it preserves the specialization of each species. In contrast, when it is reshuffled, it is used a wide utilization gradient of specialization. Furthermore, serial participation in observed matrix can be maintained or omitted. In this study, we use RA3 algorithm. This algorithm retains the amplitude and reshuffles zero conditions. In this neural model, analysis was carried out using software R and package EcoSim-R. Finally, as complement, a variance mean radio was obtained for counting data of each species to determine whether the species distribution is random if the value is 1, uniform if the value is less than 1, or aggregated if the value is higher than 1. Once the species pattern was established, it was determined if a species had Poisson, binomial, or negative distribution respectively. The analysis was done using manually Excel software. As a result, denote the presence of 5 decapod species: Cyclorhabsus cinereus, Cancer porteri, Beta-Eustrum catus, Petulis granulosus, and Molastris plana. The density values were maintained in table number 1 in this plate. In this plate, you can see at left in height Homolaspis plana, at center Petrolistes granulosus, and at the right Cyclorhabsus cinereus, whereas at low you can see at left Beta-Eustrum catus and right Cancer porteri. The result of neural models revealed that the species association were random for fixed model, whereas denoted a structured pattern for fixed equiprobable and fixed proportional simulation models that would do many repeated species by samples, whereas the result of niche sharing revealing that the species would not share ecological niche, and in consequence, it would have not interspecific competition. In this slide, you can see the result of your model analysis. And in this slide, you can see in the graphs the result of your model analysis explained in the last slides. In this slide, you can see the spatial distribution patterns observed for studied species. You can see binomial distribution for Homoraspis plana, Silurus cinereus, Beta truncatus, and Cancer porteri, and negative binomial distribution for Peroristic granulosus. These patterns will indicate that Homoraspis plana, Silurus cinereus, Beta truncatus, and Cancer porteri that have binomial distribution will have uniform pattern, whereas Peroristic granulosus that has negative binomial distribution will have aggregated pattern. As discussion, the exposed result revealed that the species composition is similar to the description to exposed littoral coast in northern Patagonia and north of Chile, where the species occurrence revealed random presence that its consequence will reveal the absence of species interactions. This result will be supported by nature-challenging null models obtained in the present study. Nevertheless, the result based on size overlap null models on interstitial dipole in central Chilean coast and two different sites in exposed coast in northern and southern Chile revealed that intertidal decapod communities have size overlap and in consequence will share ecological nature that in first instance will not support the result obtained in the present study. The result of negative binomial distribution that revealed aggregated pattern will be protective strategy, whereas the result of a binomial distribution will have due to territorial behavior, both patterns were similar to observation for the North Chilean intertidal decapod. A possible cause that would explain this contradiction between null model results for explaining intertidal decapod community would be probably the difference in oceanographic characteristics, mainly in terms of substrate conditions that will provide shelter for intertidal decapod. The result obtained in the present study will reveal that will be necessary more study for understanding interspecific interaction between intertidal decapods along Chilean coast. In this plate is the acknowledgement section. Thank you. And in this plate are mentioned some important references for this study. And in this place are mentioned more important references for this study. Thank you for your attention.
Thank you for Dr. Patricio for your valuable talk. Now we are going to move to our next speaker, Dr. Marwa Bokarma from University of Souk Ahras, Algeria. She will talk on a comprehensive examination of post-fire floristic diversity and ecosystem health in traffic pine forest. Please, Dr. Marwa.
Hello everyone, my name is Marwa Boucharma and I am a PhD student at Mohammed Sheikh Saadiya Sugaras University. Today I'm pleased to present a short communication on a comprehensive examination of forest fire floristic diversity and ecosystem health in the Tifesh pine forest. First of all, Algeria is one of the countries in the Mediterranean basin where the issue of forest fires is particularly pressing due to its devastating impact. The pine forests in the Sugaras region have been severely affected by wildfires in recent years, so to understand the fate of burned communities, it is important to monitor their evolution over time and contribute to advancing knowledge about the effects of wildfires on forest vegetation in Algerian forests, particularly on Aleppo pine stands. So our objective is investigates the effects of forest fires on plant biodiversity and to assess the recovery and dynamics of vegetation associated with Aleppo pine. Our study area is located in the Tifesh forest, is a pine forest in the wilaya of Sugaras in the northeastern part of Algeria. We surveyed a total of 9 sites, 5 that have never been affected by fire, 4 post-fire sites, and 5 recently burned sites, all of which share similar habitat characteristics. About the fire trips were conducted during the following period. The phytosociological surveys were carried out based on minimum area of 100 meters square, a plot of 10 meter by 10 meter. About the data analysis, we use species richness, biological type, morphological type, and chorological type. So, 3 states were identified. State 1 included the surveys about the data analysis that has not been a bird control. State 2 corresponds to vegetation that was recently burned 2 to 3 years ago, and state 3 refers to vegetation that has been burned for 4 to 5 years post-fire. The result at the conclusion of this floristic, ecological, and phytosociological study conducted in the Tifish forest, 70 species were recorded belonging to 4 classes, 20 orders, and 61 genera and 28 families. About the first figure shows the distribution of families by species in our study area, with the most represented families in order of importance being Asteraceae with 90 species and Lamiaceae with 5 species. The moderately represented families are Brassicaceae and Apiaceae, each with 4 species, followed by Plantaginaceae, Oleaceae, Fabaceae, and Cetaceae, each with 3 species. The observation of figure 2 shows that floristic richness is maximal in state 2 with 41 species. This richness decreased by half with 24 species recorded in state 3. And finally, state 1 contains only 18 species. From the result of analysis of biological, chorological, and ecological trees, we have 2 biological types dominate the vegetation in our study area: Hemiocryptophyte with 27 species and Pteriophyte with 24 species. Mediterranean species dominate our study area with 33 species. Western Mediterranean and cosmopolitan species rank second and third with 10 species and 9 species, followed by Eurasian species with 6 species. According to the figure of morphological type, the herbaceous plants are the predominant with 58 species. And finally, among the 70 species recorded, we have 59 species are classified as of list of concern, LC. 9 species are not evaluated. 1 species is vulnerable and 1 species is endangered. So in this slide, I would like to share with you some picture or photos of species that I recorded in our study area. And finally, conclusion. For species richness, we recorded a difference between the 3 habitat types: controlled, recently burned, and post-fire. Species appear gradually. They are few in number during the first years after the fire. Then floristic richness reaches its peak before subsequently decreasing and stabilizing in the later stages. The generally high number of species in the early years can be attributed to the opening of the vegetation cover created by the passage of fire, the disappearance of competent species, and the mineral richness in the of the upper soil layer. And finally, thank you for your attention.
Thank you so much, Dr. Marwa. It was wonderful talk. Now we are going to move our next speaker, Dr. Meena Jamil from University of Agriculture, Faisalabad. She will present her title, Assessment of Oxidative Stress Biomarker in Labiorhita Exposed to Tannolites, a Synthetic Polycyclic Musk Compound. Please, Dr. Meena Jamil.
Assalamu alaikum. Dr. Mina Jamil here from University of Agriculture, Faisalabad. Today I am going to present my research entitled Assessment of Oxidative Stress Biomarkers in Labiorata Exposed to Tuna Light, a Synthetic Polycyclic Musk Compound, on the forum of 8th International Conference on Applied Zoology 2025. Aquatic environments and our wild fauna are increasingly contaminated by emerging pollutants including personal care products, pharmaceuticals, pesticides and industrial chemicals. One of the major ingredients in nearly all our personal care products is synthetic musk compounds, which are widely used as fragrance carriers, replacing natural musk fragrances, which was extracted from the musk deer, and now they are synthetically used in soaps, shampoos, cosmetic detergents, and many other products. Polycyclic musk compounds. A common type of synthetic musk compounds now dominate the global market. Common polycyclic musk compounds including tunalide, which is abbreviated as AHT, and now overcrowded and overproduced yearly as 4,500 tons per year. PMCs persisted in the water, sediment and sludge due to low biodegradability and high lipophilicity. Detected in surface waters, aquatic organisms and even human tissues including blood, breast milk and urine. PMCs bioaccumulate in aquatic organisms reported in many fish species, crustaceans, birds, mammals, and their reported effects including the endocrine disruption, oxidative stress, enzyme inhibition, DNA as well as tissue damage and bioaccumulation leading to prolonged adverse impacts. Here are the environmental routes of polycyclic musk compounds including tunalide. First of all, they enter the environment by the transportation of raw material from the cosmetic industry due to the range of the products, directly and indirect input. The direct input include the disposal and wastage from the external application, and indirect including the washing and swimming like recreational activities, and they enter the rivers and all other wild environments and then cause the aquatic toxicity including in all wild aquatic animals including fish. The need for this research was due to the knowledge deficit and conservational needs. Water quality in Pakistan is declining due to untreated wastewater, direct dumping and rising pollutants including polycyclic musk compounds include all the musk and personal care products like all other fragrances and other emerging pollutants. Labiorata, a key aquaculture and food security species, which have the high consumer preference and palatability in our area, is declining under environmental stresses, yet data on polycyclic musk toxicity remain limited. Understanding polycyclic musk compounds impacts is essential for protecting and conserving Labiorata and all other the species in polluted waters. The objectives of the present research were the investigation of 96 or acute toxicity, including LC50 and lethal concentrations of tunalide for Labiorata, and determination of oxidative stress in different fish organs during chronic exposure of tunalide by assessing the activities of oxidative enzymes. Here the experimental design. Experimental location was Fish Toxicity Laboratory, Fisheries Research Farm, Department of Zoology, Wildlife and Fisheries, University of Agriculture, Faisalabad, Pakistan. Experimental animal was 90 days old Labiorata fingerlings, which was acclimatized prior to the experiment in the cemented tank, then transferred to the glass aquariums. And experimental media, all the parameters, physicochemical parameters of the media was tested and maintained regularly throughout the time period. The experiment was divided into two phases: acute toxicity and chronic toxicity. During acute toxicity, 96 or LC50 and lethal concentrations was recorded by recording the gradual mortality and then applying the probit analysis. Then during chronic toxicity, for 90 days, sublethal doses, which was 1/3, 1/5, and 1/7 of the LC50 was exposed, and the chronic toxicity was recorded. After chronic exposure of sublethal doses of tunalide, dose and time-dependent oxidative stress was assessed in fish organs including gill, liver, kidney, heart, and muscles. Fish sampling was undergoes every fortnightly, and tissue collection after the dissection tissues was collected for biochemical analysis. Antioxidant enzymes was measured using spectrophotometer including superoxide dismutase, catalase, peroxidase, and glutathione as transferase by standard method. The recorded data was statistically analyzed by using R software. Statistical differences was recorded by ANOVA and trend analysis, and LC50 and lethal concentrations was measured by using probit analysis. The recorded results revealed that median lethal and lethal concentrations LC50 and LC99 was measured, and they revealed the toxicity of tunalide increased in dose-dependent manner. As the dose increased, dose was gradually increased, started from 100 microgram per liter up to 1000 microgram per liter, and mortality gradually increased. At 1000 microgram per liter it was 100%, and 96 or LC50 was recorded at 676.10 microgram per liter. Here the probability plot showing the gradual mortality increase in the dose dependent manner. Results revealed the significant oxidative stress in the organs of Labiorhita exposed to tunalide. SOD significantly increased in all organs up to 45 days and significantly decreased up to 90 days, while catalase activity in all organs significantly decreased throughout the chronic exposure, while GST glutathione S transferase activity increased up to 30 days and then decreased up to 90 days in the dose and time dependent manner as compared to the control groups. And POD peroxidase enzyme activity significantly increased throughout the 90 days of chronic exposure, while liver showed the greatest antioxidant enzyme activity changes, followed by gills, kidney, heart, and muscle exhibiting the lowest alterations for both while exposed to musk. While performing the trend analysis to measure the treatment behaviors of the tunalide for different organs enzyme activity, here they shows the different trends. Here the POD activity, which was linear trend because it was increased throughout the 90 days, and like this other enzymes show different trends like quadratic, cubic, and more cyclic trends throughout the time period. In conclusion, results showed the significant oxidative stress induced in the Labiorhita organs due to the tunalide exposure. So, the further directions including enforcement of regulatory limits on polycyclic musk discharge into the aquatic environments, to promote the safer and eco-friendly alternatives of synthetic musk fragrances, and to improve wastewater treatments and regulations. Here the references, more cited literature. Thank you for your attention.
Thank you, Dr. Meena Jamil. It was nice talk. Now we are going to move to our next speaker, Dr. Bilal Rasool from Government College University, Faisalabad. He will present his title, Trichoderma species, effective biological control tool into Lepidoptera pest, contributing to zero pollution solution. Please, Dr. Bilal Rasool.
Bismillahirrahmanirrahim. Thanks for inviting me. My name is Dr. Bilal Rasool, and I am going to deliberate a very important pest management topic, with special reference to environment-friendly control. Trichogramma species, effective biological control tool in two Lepidoptera pests, contributing to zero pollution solutions. What is biological control? Uh, well, the term first coined by Harry Smith in relation to the biological control of insects, suppression of insect populations by native or introduced enemies. In genetic terms, a population leveling process in which the population of one species lowers the number of another. Why we should use biological control when biological control agents are expensive, labor intensive, and host specific, while chemical pesticides will cost effective, easy to apply, broad spectrums. We must shift our pest control methodologies towards biological control because chemical pesticides will implicate in ecological, environmental, and human health problems, uh, require yearly treatments, broad spectrum, toxic to both beneficial and pathogenic species. Uh, but biological control agents are non-toxic to human, not a water contaminant concern. Once colonized, may last for years. Host specific, only affect one or few species. Uh, well, little introduction about trichogramma, scientific classifications. These are eukaryotic Animalia, arthropods, phylum, class Insecta, order Hymenoptera, family Trichogrammatidae, subfamily Trichogrammatini, genus Trichogramma. These are minute polyphagous wasps, endoparasites of insects, approximately 235 recognized species worldwide. Small and very uniform in nature, range in size from zero point 0.2 to 1.5 millimeter. Trichogramma species vary in their host specificity. Adults require sugar as an energy source. Walking and short jumps are means of dispersal. Uh, they live in a variety of habitats. The main host insect orders are Lepidoptera, Hymenoptera, Neuroptera, Diptera, Coleoptera, and Hemiptera. These parasites are widely used as augmentative biological control agents. Trichogramma species. Here we evaluated three species as potential agents to control two Lepidoptera pests. Trichogramma evanescens, Trichogramma chylonus, Trichogramma brassicae. All three species have been extensively studied in other systems. The present research addressed their efficacy and parasitism potential on Agrotus epsilon and Eudocima felonia. Uh, it is necessary to determine the effect of high densities on parasitism, typically of mass rearing facilities. Mass rearing and augmentative releases are essential for effectively controlling pests in agriculture and other settings. Uh, Well, the studied host insects, the first one is Agrotus epsilon. These are polyphagous, important global agricultural and horticultural pests, cause extensive damage to numerous crops by cutting stems and consuming foliage, significant yield loss. This pest attack more than 49 plant species, making it a cosmopolitan and destructive pest of agricultural and horticultural crops globally. Its widespread presence, coupled with insecticide resistance and environmental concerns, necessitates the development of sustainable management strategies such as biological control and the use of biorational compounds. The extent of yield loss varies, but under moderate to heavily infested conditions, the damage to crop, for example, maize, can range up to 30 to 40%, and in severe outbreaks, as high as 90%. The second host insect is Eudosima felonia, known as the fruit-piercing moth, significant pest of over 100 fruit species like citrus, apple, mango, strawberries, and vegetables, feeding on ripening fruits. It causes economic damage by piercing fruits with its sharp proboscis to extract juices, leading to spoilage and infections. The economic impact typically stays below 30% in a normal year. However, outbreaks can be devastating, with losses reaching up to 100%. The expansion of fruit crops, environmental changes, rising temperatures, and reduced rainfall have disrupted population dynamics, leading to annual outbreaks. Insects with their short generation times and high reproductive rates are highly sensitive to climate variations but can quickly adapt to these changes. Although it contributes to pollination and serves as prey for predators, its primary economic impact results from the damage it inflicts on crops, necessitating effective pest management strategies, including biological control. The research objectives to select candidate species for mass rearing and eventual field release. It is essential to conduct basic research. This present research will address the following questions. Uh, how does the trichogramma wasp and host affect parasitism and emergence in both hosts? How does host species and host egg age influence wasp longevity and performance? How does the density of trichogramma wasps and hosts affect parasitism potential in both hosts? Uh, how parasites can contribute towards zero pollution solutions? Well, after conducting a series of uh, uh, experiments based on uh, choice and no choice tests, the results of parasitism are as follows. Trichogramma vansans exhibited higher parasitism than Trichogramma brassicae and Trichogramma pylonos for both hosts. In contrast, Trichogramma brassicae exhibited higher parasitism in Agrotus epsilon than Eudosima pylon. The results of emergence differences were observed among two hosts and three parasites. Higher percentage emergence was observed in Trichogramma evanescens and Trichogramma pylonos on Eudosima pylonia X, while Trichogramma brassicae showed higher emergence on Agrotus epsilon X. The experiments of longevity of parasites resulted longevity of adult parasites was significantly different depending on host parasite. Agrotus epsilon X on Agrotus epsilon X trichogramma vessels lifespan was 11.3, Trichogramma colonus 9.5, and Trichogramma brassicae 8 days. While on the Eudosima felonia, the lifespans were Trichogramma vessels 12.7 days, Trichogramma colonus 10.3, and Trichogramma brassicae 7.3 days. The experiments of parasitism and host egg age resulted the percent parasitism of all three parasites was lower on fourth day eggs, while highest on the first day old eggs. Highest parasitism was recorded on first and second day old eggs of both hosts. Uh, well, the host preference of parasites resulted among the parasites. Trichogramma evasans exhibited higher parasitism rates compared to the other two species. Both Trichogramma valencens and Trichogramma pylonos prefer two parasites, Eudosima felonia X over Agrotus epsilon, 51 to 52% versus 48 to 49% in both cases. In contrast, the Trichogramma brassicae preferred Agrotus epsilon X 52% over Eudosima felonia X 48%. Well, the experiments of host and parasite densities parasitism at increasing host and parasite density resulted host parasite density significantly affected parasitism rates in both hosts according to the relative ratios Reading chamber 1 to Reading chamber 5. The parasitism of Trichogramma vansans Trichogramma chylonus and Trichogramma brassicae significantly differed in both hosts. Trichogramma evanescens showed significant higher parasitism rates than Trichogramma chylonus, though Trichogramma brassicae showed lower parasitisms across five tested relative densities. The rating chamber 5 ratio 30 parasitoids and 900 host eggs exhibited higher parasitisms, revealing a strong positive correlation between densities and parasitisms. And the parasitisms increased with the increase of host and parasitoid densities. The conclusions, Trichogramma parasitoids were assessed for their effectiveness against two key Lepidoptera pests, Agrotus epsilon and Eudocima felonia. Both Trichogramma vaccines and Trichogramma pylonus proved effective against Eudocima felonia, while Trichogramma brassica displayed a preference for the eggs of Agrotus epsilon. These findings offer insights for developing environment-friendly pest control strategies that focus on the mass reading and augmentative releases of trichogramma targeting both species. This research highlights the importance of combining different trichogramma species and understanding their interaction with pest hosts to enhance food safety and reduce pesticide residues in sustainable agriculture, contributing towards zero pollution solutions. Well, thanks for your patience and attention. If you have any questions, please write on my email address.
Thank you very much, Dr. Birasul, for being the part of ICAZ 2025. Now we are going to move our next speaker, Dr. Sahresh Firal from University of Veterinary and Animal Sciences, Lahore. She will present macrolide resistance in Streptococcus aureus associated with bovine mastitis and improvising the molecular docking as an alternative treatment. Please, Dr. Sahresh Firal.
It is a pleasure to be here today to present my research on macrolide resistance mechanism in Streptococcus aureus associated with bovine mastitis, focusing on the ERMB and MEF-A genes and exploring the alternative treatment options through molecular docking. Bovine mastitis remains one of the most economically significant diseases in the dairy industry, affecting the milk production and animal health. Among the bacterial pathogens responsible, Streptococcus aureus plays a major role. While macrolide antibiotics are commonly used to treat infections, the increasing emergence of resistance, particularly mediated by ERMB and MEF-A gene, poses a serious challenges. In this study, I aim to analyze the genetic basis of macrolide resistance in Streptococcus aureus isolates and investigate the potential therapeutic or alternative therapeutic compound using molecular docking techniques, and by identifying novel molecular interactions, I hope to pave the way for more effective treatment strategies that can overcome antibiotic resistance. In the next few minutes, I will walk you through the methodology, key findings, and potential implications of this research, particularly in combating antimicrobial resistance in veterinary medicine. The effects of mastitis include the decreased milk production, and the most significant and costly infection in the dairy sector is mastitis. Due to decreased milk production, it causes significant financial losses, productivity, higher labor costs and treatment costs, and milk withheld after treatment. Overall, the prevalence of clinical and subclinical mastitis was 17 and 57% in both species. Clinical mastitis was higher in cattle, 20 to 21%, and compared to buffaloes, 11%, whereas subclinical was higher in buffaloes, about 66%, than cattle, 53%. Besides, month-wise prevalence was higher in hot and humid months in both species. In Pakistan, prevalence of the subclinical mastitis is 17 to 93% in cows and 4 to 48% in buffaloes. The dairy industry is facing a great setback due to high prevalence and incidence of the mastitis in milk animals. The infection rate of mastitis in cows with pendulous udder is higher than those with non-pendulous udders, and the pendulous udder exposes the teeth to injury, and the pathogens may easily adhere to the teeth and get access to the gland tissues. The infection rate in cows with teeth lesions is more than cows with normal teeth. The organisms involved in mastitis vary from community to community. The most common causative organism of udder disease include Staphylococcus strains S. aureus and S. epidermidis, Streptococcus strains A. galacti and Dysgalacti and Ubarus and bovis, and coliforms mainly E. coli and Klebsiella pneumonia. Other less frequent agents include Pseudomonas nocardia and Streptococcus and E. coli and Pseudomonas are found in buffalo suffering with mastitis. This pie chart representation shows prevalence of Staphylococcus species, and that is about 47%, and Streptococcus species is 21%, and E. coli species about 18%. Streptococcus uberus is considered environmental strain responsible for deterioration of the infected quarters in dairy animals. Aggressive inframammary antibiotic regimens, including use of macrolide, is one of the reasons to impart antimicrobial resistance. Alternative medication against this bovine mastitis is essential for current state of mastitis to reduce the implementation of antibiotics. So Streptococcus uberus is mostly found in the environment of the dairy farms, and general areas of growth of the strain include the water and soil. Here are the objectives. Molecular identification of macrolide resistance of ERMB and MEF-A gene in Streptococcus uberus, and to find out the alternative treatment for bovine mastitis by improvising the molecular docking as an alternate treatment. The research work was carried out in postgraduate laboratory IBBT UAS. Total of 20 milk samples from the mastitic cows were collected from different farms, and for the confirmation of mastitis, subfield mastitis test was performed at the site. And subfield mastitis test was performed by mixing the 3% detergent solution with the milk in equal proportion. Blood agar media was used for the isolation of the Streptococcus uberus, and gram staining was performed for phenotypic characterization. Macrolide resistance activity by Streptococcus uberus was determined by the implementation of disc diffusion method, and erythromycin and azithromycin discs were used. DNA from macrolide resistant Streptococcus uberus strains was extracted by using the organic method, and extracted DNA was quantified by that of the nanodrop spectrophotometer. Amplification of ERMB and MEF genes was performed by PCR. For this purpose, two sets of primers were used. On sequencing results, molecular docking was performed. This figure represents the bacterial isolation and identification was performed on blood agar plates after the incubation of 48 hours at 37 degrees Celsius. Streptococcus uberus growth appears as a result of primary and secondary culturing. The colony is small, moist, and translucent and slightly whitish. For characterization, phenotypic analysis was performed. Colony morphology was studied by gram staining. The slides were viewed under a microscope. Here is the result of gram staining. Appears the gram positive bacteria appeared as cocci in small chains. Macrolide resistance activity by Streptococcus uberus was determined by the implementation of disc diffusion method. 15 microgram of erythromycin and azithromycin discs were used on Muller-Hinton agar media. Streptococcus uberus is treated with the macrolide, but this strain is resistant to this common class of antibiotics, including azithromycin and erythromycin. The reason behind this resistance is the presence of MEF-A and ERMB genes in Streptococcus uberus. The resistance caused by MEF-A gene is considered M-type resistance against the macrolides, and this gene is also considered the secondary transporter of the various compounds. Other genes involved in macrolide resistance include the ERMB gene that encodes the enzyme termed as ribosomal methylase, and it is the major concern with the perspective of the prevalence of mastitis caused by Streptococcus aureus. And zone of inhibition was measured in mm, and erythromycin shows the measurement of the 12 mm and azithromycin shows the 17 mm, and results were interpreted according to the guidelines of the clinical laboratories and standard institute. For molecular identification, DNA extraction was performed by the organic method from the overnight incubated LB broth of the cultured colonies, and figure A represents the bands extracted by extracted DNA visualized on the 0.8% gel by using the BioRed gel documentation system, and figure B represented the graphical representation that shows the purity and concentration of extracted DNA. So the optical density was calculated at ratio 260 by 280. This ratio was used for the purity of the DNA. This figure represents the PCR amplified products of MEF-A and ERMB genes, and those amplified products of 346 base pair and 203 base pairs were visualized on 2% agarose gel by using UV transilluminator, and DNA letter was run in the first well. This figure this shows the representation of the sequence, nucleotide sequence. The detailed sequence obtained after the sequencing of the confirmed Streptococcus aureus strain is provided in a FASTF format. Results of sequencing was converted into the protein sequence. Translation were performed on the nucleotide sequences of the Streptococcus aureus genes obtained as a result of the sequencing by using the online tool ICAZ-2025. ICAZ-2025 translated the nucleotide sequence into the protein sequence, and after that the sequence was converted into the nucleotide sequence and into the tertiary form. The ProCheck server was used to confirm the protein's tertiary structure, and the protein tertiary structure is in the form of the PDB. Then the ProCheck server converts the PDP structure into the Ramachandran plot, and ProCheck system to verify the tertiary structure of the protein. The validation findings were given by the Ramachandran plot, whose data show that the residues in the most favored areas for MEF-A gene and the ERMB gene and MEF-A gene around 91% residues in the further permitted regions, whereas approximately 6.7% residues in the disallowed regions were approximately 1.1%, and those residues in the generously allowed regions including 1.1%. There were around 100 residues in all. Phi and psi stand for the degrees. Red color indicate the residues in most favored regions, yellow color indicate the residues in additional allowed regions, and white color represented the residues in disallowed regions, and residues in generously allowed regions indicated by light brown color. Total of 20 phytochemicals were chosen for the screening of the most appropriate phytochemical mechanism model. The ST format structure of those ligands were given by PubChem. However, the ST format is converted into protein data bank format for further processing. All of the phytochemical medications contain properties that make them effective against the pathogenic protein. Total 20 selected phytochemicals were piperine, berberine, dictamine, sanguinarine, canoclavin, chylithrine, metrine diallyl sulfate, and the reversitol, allicin, 3-hydroxyflavone, carvacol, thymol, and coumarin, codeine, agilinol, and osthol, and cinnamaldehyde, and eugenol, and mescaline. Multiple docking during a single run on Pyrex produce a score of the -6.4 and -6.5, and the best phytochemical scoring model. Out of 20 phytochemical compounds, sanguinarine and mescaline were chosen. Based on the rating of that explain the binding affinity, sanguinarine and mescaline were chosen. Additionally, the phytochemical diallyl sulfide has the lowest binding affinity score at around -2.6, the result of the grading 20 chosen phytochemicals. The ERMB and MEF-A gene confer macrolide resistance in Streptococcus aureus, reduces the efficacy of traditional therapeutics and use of phytochemicals effective therapy for bovine mastitis. So here is the conclusion. Antimicrobial resistance activity of Streptococcus suberis against macrolide was identified that reduces the use of erythromycin and azithromycin as medication against Streptococcus suberis. Macrolide resistance was facilitated by the ERMB and MEF-A genes. In silico approach provided the effective phytochemical for the treatment of bovine mastitis. So the phytochemical can be used in place or in addition to conventional antibiotics. Prediction of the suitable phytochemical as therapeutic against bovine mastitis by the implementation of bioinformatics that reduces the stress on animals. Thank you. That's all from my side.
Thank you very much, Dr. Sarish Fral. It was wonderful talk. Now we are going to move our next speaker, Umme Habiba from Women University, Multan. She will present in vivo anti-lipidemic and anti-hepatotoxic evaluation of Origanum majorana against dexamethasone. Please, Umme Habiba.
Assalamu alaikum. I am Dr. Umme Habiba from Department of Biochemistry and Biotechnology, the Women University Multan. And here I am presenting in 8th International Conference on Applied Zoology 2025. The topic of my presentation is in vivo antilipidemic and anti-hepatotoxic evaluation of Origanum marijuana against dexamethasone. In this study, we use liver. We work on hepatotoxicity because liver is the second largest organ of the body, and liver support immunity, digestion, metabolism, detoxification, and transformation of drugs and metabolites. It also maintains cholesterol homeostasis and store fat-soluble vitamins and minerals. Different factors are involved that cause hepatotoxicity. In these factors, different viruses, exogenous substances like drugs, alcohol, radiation, high-fat diet, ischemia, sepsis, in which bacteria or any type of viral infection, and autoimmune disorders are involved that induce in hepatotoxicity due to the increase of the amount of reactive oxygen species. And in case of increase of reactive oxygen species, lipid peroxidation in the lipid hepatic cells increase that harm the cell membrane and weaken the antioxidant defense system and also attack the nucleophilic residuals of the DNA. In this study, we used dexamethasone that is a commonly used drug, synthetic glucocorticoid, to induce the hepatotoxicity because its high dose affect naturally occurring substances and increases triglyceride lipid level in the body and also induced fatty liver, and it's also caused the high production of reactive oxygen species when its high amount is taken. And to cure this and we overcome the reactive oxygen species by using Origanum majorana. Because Origanum marijuana belongs to the class of Lamellaceae family and its common name is oregano. It has strong antioxidant qualities which enable it to hunt free radicals and shield cells from oxidative stress-related destructions. It has also anti-diabetic effects, increasing glucokinase activity and promoting the production of liver glycogen and also elevate the plasma insulin level. So in this study, we used Origanum marijuana plant, and firstly, we taken this plant from university botanical garden, and then the plant was dried, and then their powder was used for the maceration process, in which acetone was used as a solvent, is used for solvent extraction and as a solvent extraction, and then at the end acetone extracts were prepared, and these extracts were used to find out their phytochemical analysis or in which total phenolics and total flavonoid contents of the plants were measured. After that, in DPPH radical scavenging assay was used to measure the percentage inhibition of DPPH that is used as an antioxidant agent. And for in vivo analysis of plant extracts, firstly weigh the rats and then divide it into 5 main groups: control group, positive control, negative control, curative, and standard group. And then different and those was decided on the base of the rats of body weight of the rats, and plants were given, and in case of curative treated group, and resveratrol that is used as a standard drug for hepatoprotective as a protective agent. It was used as a standard drug to compare the effects of our plant and with standard drug. The whole time period was 21 days in which plant was administered, and firstly blood was taken at 0 day, and then at 3rd day to check the hepatotoxicity of dexamethasone, and then at 10th day and 21st day, and at the end of the experiment whole period, then we take liver organ, remove the liver organ from dissected rats to perform the histopathology. And blood samples were analyzed as a liver function test and for lipid profile. The results of in vivo analysis indicated that total phenolic contents 17.43 and total flavonoid contents 17.43 and total phenolic content 5.78 were observed at concentration of 2.5 milligram per ml, and maximum percentage inhibition of DPPH was measured at was observed at 57.545 at 2.5 milligram per ml concentration of the plant. And the in vivo results in rats indicated that at 0 day, normal liver enzyme ALT and AST were present in all rat groups, while at 3rd day in both cases of ALT and AST, and increase of ALT level was AST level was observed in case of positive control group due to the administration of dexamethasone. And in case of plant treated and drug treated groups, at 10th day and 21st day, normal level of ALT and AST were observed in treated and standard groups. Similar findings were obtained in case of alkaline phosphatase, gamma glutamyl transferase, and albumin. In case of lipid profile, therapeutic effects of marijuana on lipid profile, cholesterol, triglycerides, HDL, and LDL level were measured, and they indicated that in case of positive control group that was treated with dexamethasone show high level of cholesterol, triglycerides, HDL, and LDL as compared to treated group in which slight increase in the lipid profile was observed. And these are the results of histopathology. In control group, intact hepatolobules, central vein, hepatolobules, central vein, and sinusoidal dilation. The clear cell membrane was observed. Right? Sorry. In case of positive control group that was treated with dexamethasone, show inflammation, necrosis, and hepatic parenchymal cells inflammation, while in case of treated group that was firstly treated with dexamethasone and then so with plant or marijuana show positive results in which mild inflammation and edema was observed in plant treated group. And in case of standard group, as you can see, mild necrosis and dilation of veins were observed as compared to positive control group. It means our plant or marijuana show positive histopathological results and show that it combats with disease, hepatotoxicity, and plants show potential against hepatotoxicity. So from the study, we concluded that oregano marijuana potential as a homeopathic treatment for reducing an elevated cholesterol level and hepatotoxicity that is caused by dexamethasone. And oregano marijuana has a great promises in preventing oxidative damage to liver cells, reducing inflammation and controlling lipid metabolism because of its anti-inflammatory, hepatoprotective, and antioxidant properties. And these are the future perspectives. So, for more clinical trials required to confirm the efficacy of marijuana in human subjects. And also, marijuana may have more medicinal promises if standardized extracts or formulations with optimal doses were developed. And oregano marijuana natural origin and diverse pharmaceutical properties make it a promising choice for upcoming therapeutic approaches that protect liver health and maintain metabolic balance. These are the references. Thank you for your attention.
Thank you very much, Habiba. Now we are going to move our next speaker, Ms. Rabia from University of Agriculture, Faisalabad. She will present her title, Effect of Induced Bisphenol A Toxicity on Histopathology of Sarhinus mirgala. Please, Ms. Rabia.
Assalamu alaikum everyone. My name is Rabia and I am MPhil scholar from University of Agriculture, Faisalabad. Today I am excited to present my research topic, Effect of Induced Bisphenol A Toxicity on the Histopathology of Sarhinus mirgala. My research focused on evaluating the histopathological effects of induced bisphenol A toxicity on vital organs, gills and muscles of Sarhinus mirgala, a widely cultured freshwater fish species. Then we will discuss about the introduction of bisphenol A. BPA is a significant chemical compound primarily utilized for the manufacturing of epoxy materials and polycarbonates. It is a non-endocrine disrupting compounds that enters aquatic ecosystems through industrial discharge and plastic waste. In addition to its use in thermal paper manufacture, BPA is used as antioxidant and stabilizer in vinyl chloride synthesis. The synthesis of BPA was initially achieved in 1891 through the condensation of phenol and acetone. BPA is recognized as chemical. BPA is detrimental to fish in the aquatic environment and extended sites wastewater treatment effluent or natural degradation process. The main objectives of my research: assessment of the acute toxicity LC50 and lethal concentration of BPA for Sarhinus mirgala, examination of the effects of induced bisphenol A toxicity on the histopathology of Sarhinus mirgala. Then we will discuss about the methodology. The experiment entitled Effects of Induced Bisphenol A Toxicity on the Histopathology of Sarhinus mirgala was conducted at the University of Agriculture, Faisalabad. The fish sample of the Sarhinus mirgala were collected from the University of Fisheries Research Farm. Fish fingerlings of the same weight and length were selected for the experiment. The fish were acclimatized for 10 days in glass aquarium having 50 liters of aerated tap water. Dead fish and fish showing behavioral changes were discarded daily. The water in the aquarium were replaced readily with fresh water. Then physico-chemical parameters such as temperature, DO, pH were maintained at optimal levels throughout the experiment. BPA was dissolved in DMSO solvent and its stock as well as working solution were prepared. BPA concentration started from 1 microgram per liter and gradually increased up to 10 microgram per liter for acute toxicity testing. The 96 odds, LC50 and lethal concentration of BPA were also measured. 3 replicates were used to assess the toxicity testing of lethal concentration of LC50 of BPA for Sarhinus mirigilla. After determination of lethal concentration, sublethal doses of BPA were used to observe histopathological changes in different organs of fish to check chronic effects. Fish were dissected after exposure and organs such as muscle and gills were separated and preserved in 15% neutral buffered formalin. Then the slides were prepared and studied under a microscope. Statistical analysis use correlation and probability analysis. Then we will discuss about the results of my research. Histopathological alteration induced by the BPA in the exposed organ gills of the Sarhinus mirigilla. Histopathological examination of the fish species Mori of the control group at 10x and 40x object lengths under the light microscope showed no significant changes in the fish gills tissues. Low dose of BPA showed histopathological examination of the fish species Sarhinus mirigilla showed low level significant alteration in the fish gill tissues. Well-defined primary and secondary lamella, unhealthy epithelial cells, low level of cellular damage. Then medium dose of bisphenol A. Histopathological examination of the fish species of Sarhinus mirigilla treated with BPA showed and significant structural changes were observed in the gill tissue. Fish treated with BPA showed more substantial structural alterations including epithelial lifting, lamellar fusion and moderate necrosis in some regions. Then high dose of BPA. Histopathological examination of fish species Sarhinus mirigilla treated with BPA showed a highly significant structural alterations were observed in gill tissues. BPA showed more structural alterations including epithelial lifting, lamellar fusion and moderate necrosis. Then histopathological alterations induced by bisphenol A in the exposed organ muscles of Sarhinus mirigilla. Histopathological examination of the fish species Sarhinus mirigilla of control group showed no significant changes in the muscle tissues, no signs of degeneration and no signs of any type of cellular damage, clear striations, well-defined muscle fibers. Low dose of BPA showed histopathological examination of fish species Sarhinus mirigilla showed low significant changes in the muscle tissues. Muscle fibers, clear striations, sign degenerations and others. Moderate dose of BPA showed histopathological examination of fish species showed with BPA significant structural changes were observed. High doses of BPA showed histopathological examination of fish species Sarhinus mirigilla treated with BPA, more substantial structural alterations and muscle injuries due to toxicity. These results showed that BPA can cause significant fish muscles tissue damage as compared to control group. Then discuss the conclusion. LC50 and LC99 were measured for Sarhinus mirigilla as 5.85 plus minus 0.41 microgram per liter and 12.37 plus minus 1.18 microgram per liter respectively. The gills and muscles tissue of the Sarhinus mirigilla fair damage as compared as a result of exposure to BPA. All physicochemical parameters were significantly correlated with BPA concentrations. The population of Sarhinus mirigilla is decreasing as consequence of of habitat loss, regeneration, degradation, water obstruction, pollution, and eutrophication. This study concluded that BPA chemicals are highly toxic to fish. BPA affect fish. BPA causes hormonal imbalance, reproductive system disorder, developmental behavioral changes in fish. Thank you for your attention.
Thank you very much, our young researcher. Now we are going to move our next speaker, Miss Arwa Noor from University of Agriculture, Faisalabad. She will present on studies of histopathological alteration induced by chronic exposure of bisphenol A in hypophthalmitis mollitrix. Please, Arwa Noor.
Assalamu alaikum everyone. My name is Arwa Noor and I am an MPhil scholar from University of Agriculture, Faisalabad. Today I am excited to present my research study of histopathological alterations induced by the chronic exposure of bisphenol A in hypophthalmitis mollitrix. My research focused on evaluating the histopathological effect of induced bisphenol A toxicity on vital organs like gills, muscles of hypophthalmitis mollitrix. Now I will discuss about the introduction of my research. First of all, what is BPA? BPA stands for bisphenol A. Bisphenol A is a chemical produced by the combining of two phenol groups and one acetone molecule. Bisphenol A is widely used in the production of medical equipments, tooth fillings, lens coatings, optical and lenses, toys, storage media, and window foils. BPA is a persistent pollutant in the aquatic environments because of its presence in sewage from cities, landfill runoff, and the petrochemical industries. Bisphenol A is present in water bodies and its release has a substantial effect on the growth and the development of fish by affecting energy metabolism, individual cortisol stress reaction, axon growth, muscle mass, and motor behavior. Bisphenol A is also harmful to aquatic life and environment. Aquatic animals are also in grave danger from this kind of exposure. Now I will discuss about the objective of my research. Assessment the acute toxicity of bisphenol A in hypophthalmitis mollitrix by determining the lethal and sublethal concentrations. Evaluation the histopathological alterations induced by the chronic exposure of bisphenol A in silver carp. Now I will discuss about the materials and methodology of my research. To evaluate the effect of bisphenol A on histopathological of different organs of silver carp, my research was conducted at the Fisheries Research Farm, Department of Zoology, Wildlife Fisheries, University of Agriculture, Faisalabad. Fish were acclimatized for 10 days in glass aquarium using 50 liters of dechlorinated and aerated tap water. Physicochemical parameters of the water like optimum temperature, dissolved oxygen, DO, pH, total hardness, carbon dioxide, calcium as well as magnesium were also checked during the experimental trial. BPA was dissolved in the dimethyl sulfur oxide solvents and its stock as well as working solution were also prepared. 3 replications were established to investigate the toxicity of bisphenol A on hypophthalmitis mollitrix. 10 fish were kept in each replicate. BPA concentration started from 1 microgram per liter and gradually increased up to 5.5 microgram per liter for acute toxicity testing. 96 or LC50 and lethal concentrations of BPA were also measured. Probit analysis method was used to calculate the LC50 and lethal concentration of BPA. Sublethal doses 1/7, 1/5 and 1/3 of LC50 of BPA were also used to observe histopathological changes in different organs of fish like gills and muscles. Fish were dissected to check the histopathological alterations in different organs of the fish such as gills and muscles were separated and preserved. Statistical analyses like correlation and probit analysis were also observed. Now I will discuss about the results of our research. Histopathological alterations induced by the bisphenol A in the exposed organs gills of the hypophthalmictus mollitrix. Histopathological examinations of the fish species silver carp of the control group showed no significant changes in the fish gill tissues, while low dose of bisphenol A showed histopathological alterations of the fish species of the control group showed low significant alterations were examinations in the fish gill tissues, like well-defined primary and secondary lamellae, healthy epithelial cells, and an absence of the cellular damage was observed in the silver carp fish gill tissues. Although medium dose of bisphenol A showed histopathological examination of the fish species treated with bisphenol A showed a significant structural change were observed in the gill tissue. Fish treated with bisphenol A showed more substantial structural alterations, including epithelial lifting, lamellar fusion, and severely necrosis in some regions. Although high dose of bisphenol A, 1/3 of the LC50, showed histopathological examination of fish species silver carp treated with bisphenol A showed a significant structural alterations were observed in the gill tissues. The fish treated with bisphenol A showed more substantial structural alterations, including epithelial lifting, lamellar fusion, and necrosis in the regions of the gill tissue. Now I will discuss about the histopathological of hypophthalmictus mollitrix exposed to the muscles. Histopathological examination of fish species like silver carp of the control group had showed no significant changes in the muscle tissue, while low dose of bisphenol A showed histopathological examination of the fish species hypophthalmictus mollitrix of the control group showed no significant damage in the muscle tissue. Although medium dose of bisphenol A showed histopathological examination of the fish species silver carp treated with bisphenol A showed a significant structural changes were observed in the fish muscle tissue. Histopathological examination of the fish species treated with high dose of bisphenol A at the concentration revealed significant structural changes in the muscle tissue, like including muscle fiber fragmentation, increase in interstitial gaps, denaturation, and the reduction in the number of nuclei in the muscle tissue. Now I will discuss about the conclusion of my research. LC50 and LC99 were measured for H hypophthalmictus mollitrix as 3.67 plus minus 0.21 microgram per liter and 6.98 plus minus 0.64 microgram per liter respectively. Exposure to BPA caused significant damage in the gills and the muscle tissue of the hypophthalmictus mollitrix. All physicochemical parameters were significantly correlated with BPA correlation. Conclusion of my research revealed that the exposure to BPA caused a significant damage in these organs, such as damage in mucous, epithelial cells of the gills, and the muscle fragmentation. These changes include cellular degeneration, necrosis, hemorrhage, and inflammation, all of which indicate toxic stress and impaired organ function. The severity of the tissue damage was found to correlate with both the concentration of BPA and the duration of exposure, highlighting the dose-dependent toxic effect of this endocrine disrupting chemical. Thank you for your attention.
Thank you very much, Arvanur. It was nice presentation. Now we are going to move our next speaker, Taiba Rani from University of Sialkot. She will present her talk on the title Estimation of Heavy Metal Accumulation in Indian Pond, Huron from the district Narwal. Please, Taiba Rani.
Rahim. Hi everyone, my name is Taibarani and I am MS Scholar of Zoology at University of Seattle. First of all, I would like to thank you to the amazing organizing committee on 8th International Conference on Applied Zoology, ICAZ 2025, for giving me opportunity to share my thoughts with you on this amazing forum. The title that I'm going to present here is The Estimation of Heavy Metal Accumulation in Indian Pound Heron from District Narwal. Here are table of contents. Number 1 is introduction, objective, materials and methods, results, conclusion and references. If we talk about heavy metals, those metals that have their atomic number greater than 20 and having density greater than 5 gram per centimeter cube are considered as heavy metals. Cadmium, lead, mercury are one of these examples. These metals are dense and non-biodegradable elements and having their property to accumulate into the body of living organism and causes a multiple group of diseases like GI tract diseases, respiratory diseases, cardiovascular diseases, reproductive renal disease, hemopoietic and neurological disorders. This is the species that I have selected for my research study. This aquatic species, which is Indian pound heron, belonging from the family that is RDD, where 120 species of this family are found across the world and 9 species reported in Pakistan, and Indian pound heron is one of them. The reason for selecting this species is it is a least concerned species globally and in Pakistan according to the red list by IUCN 2024. Main sources of the metal pollution in environment are natural and artificial means. Through natural means, weathering of rocks and volcanic eruptions are included, and in artificial means, different types of pesticides, fertilizers, industrial waste are included. These three main pathways through which a bird get uh, accumulated these metal pollution. These are soil, water, and air, and getting these metals by different means, that is ingestion or eating, drinking, and inhaling. The objective of my research study is to detect the heavy metal in feather, kidney, and liver. And number 2 is to evaluate the difference of heavy metal concentration from different habitat type of district Narwal. So next is a study site. This is the map of my study sites. The district Narwal consists of 3 tehsils: Tehsil Zafarwal, Tehsil Shakargarh, and Tehsil Narwal. The experimental overview consists into 3 main steps. Number 1 step is sampling. Number 2 step is digestion. Number 3 step is detection and analysis. This is the total sample size and sampling area. The district Narwal divided into three sites, that is site 1, site 2, and site 3. The total sample size is 27. Sampling and digestion of feather is performed into different steps. Number 1 is collecting the samples, then washing the sample. After that, dry into oven drying. Then digestive reagent are used for digesting the feathers. Then complete digestion, we use hot plate. After that, filtration and dilution are performed for analysis. Sampling and digestion of organs, that is liver and kidney, are performed through the same steps, but there is one acid, that is hydrogen peroxide is used instead of perchloric acid. This is the technique, that is inductively coupled plasma. Optical emission spectroscopy is used for detecting the metal. For statistical means, we use RStudio and apply a test, that is one-way ANOVA test. Results. If we talk about the results, there are 3 main organs, that is feather, kidney, and liver are used in this research, and the 3 main study sites, site 1, site 2, and site 3. Site 1 is Narwal, Site 2 is Shakargarh, and Site 3 is Zafarwal. The summary of my research study is these are three different, and there are multiple sources of pollution, and these are get entered into the body of a bird by different means and get accumulated into the organs. When these organs are digested and analyzed by ICPOES, then the detected metals are statistically applied by our studio. This is the logarithmic bar graph where different colors of lines are shown, lines and dot are shown. So number one, highest peak is green line, and followed the red line and then blue line. If we talk about the legends, the legend of organ shows that the green line shows the kidney, and red to the feather, and blue to liver. It means that kidney and feathers get more accumulated the metal than liver. This is a logarithmic line graph showing the association between metals and to the sites. Here again shown different sites, 3 sites and 12 metals. These 12 metals associated with these 3 sites, but shown the red line to above, follow the blue line, and then to green line. If we talk about their peaks, the highest peak is 1000 plus, which is follow the site 1, and after that site 3, and the lowest one is site 2. It means that kidney and liver, kidney and feathers accumulated more metals than the liver, and nickel shows the highest peak in site 1 and site 3 than in site 2. That's all from my side. Thank you for your kind attention.
Thank you very much, Taibarani. First session is going to end. Now we are moving to our next session, which will be moderated by Ms. Samia Zahoor. Please stay tuned and stay connected. Thank you.